Good morning to all of you. Today I am here to tell you about some useful and harmful bacteria, also the structure of bacteria. So if you will talk about the structure of bacteria, you can see here so many things are there present. Capsule is there, cytoplasm is there, flagella is there, cell wall is present, plasma membrane is present. Now we will come to the functions. So first function, capsule is preventing, that is the outermost structure, and it is protecting the bacteria from outside effects. Second is the cell wall, that cell wall is also present, that is responsible for protecting the inside contents from the outside environment. Then you have plasma membrane that is acting as a sheath for the inside components, that is not letting it to harm in the internal components of bacteria. And they have the cell wall, it is covered by the cell wall also. So and then it has cytoplasm, where all the different organelles, if present, it will be present over in the, it will be dissolved in the cytoplasm. Then you have flagella's work is only to help the bacteria in the movement. So if flagella is more, it is less in number, and if cilia is there, it is less in number. So cilia and flagella, these are the locomotory organs in prokaryotes, where you can see this flagella will be small in number. So here you see only 3 flagella are there, but if cilia will be present, might be present, then it will be 100, 200 will be present. Okay. So as a whole, this is about the structure of bacteria. Now we will come to the useful effect of bacteria. So the first useful effect is rating. So what is rating? Rating is the jute fibers when they are obtained from the plant. So to separate the stem and the fibers, they are attacked by, they are exposed to bacteria, and then what will happen? The stem and the bacteria, they will be easily separated from the stem. Okay, the fibers of the plant, they will, when they will be acting upon by bacteria, they will be acted upon by bacteria, then the fibers and the stem will separate. This is called rating process. Now come to curing. Curing is the process in which tea and tobacco, or whatever, tea and especially tea and coffee, they are cured, or they are exposed to bacteria so that a special flavor can be given to them. That is called curing. Okay. So the taste, whatever the flavor, whatever is present in the coffee leaves, tea leaves, that is only due to the bacteria. Next is setting of curd. Everybody knows milk is, when milk is converted to curd, then milk, this is setting of curd is due to lactobacillus bacteria. That is bacilli, that is a type of bacilli. Okay, lactobacillus. Everybody is knowing about this setting of the curd. Then it is tanning of leather. Tanning of leather, what is that? The hides, when the hides are separated, to make the, when the hides are, will be exposed to bacteria, then you get the, it is converted, hides are converted into leather. That is, this process is called tanning. Okay. Next is your vinegar production. So for the production of vinegar, acetobacter is the bacteria which is responsible for vinegar production. Vinegar, what is that? Acetic acid. Vinegar is also called acetic acid. Everybody in the kitchen also vinegar is present. Most of you might have observed what is vinegar. If you have not seen, you go to the kitchen and see what is vinegar. Okay. In some of the fast food also, chow-mein and all, it is given, or it may be used to produce the pickles. Okay. In the home, you might have seen this vinegar. Okay. Next is your fuel production. So production of the fuels also, for the production of fuels, bacteria are necessary. Okay, the fuel, just like the CNG and all, to extract all this, you need some bacteria. Then sewage treatment. So the treatment of sewage, the solid wastes, they are treated by the help of bacteria. The primary, secondary and tertiary treatment. So the treatment generally without the bacteria, it is not possible, because bacteria has the capacity or capability to digest all the different non-useful components of the atmosphere to convert it into useful component. Okay. Then it is nitrogen fixation. So nitrogen fixation, it is a process where what is happening in the leguminous plants which are not able to take the nitrogen directly from the atmosphere. This bacteria, it is living in with the root hairs. Okay, symbiotic association, it is living with the root hairs, and that is responsible for fixation of nitrogen, and this nitrogen is converted into nitrates, nitrites, so that it can be properly used by the plants. So this is another effect, nitrogen fixation of, it is a useful effect of bacteria. Then compost. Compost is the manure which is formed from the, just like the biogas, whatever is being treated, it is also a type of compost. And compost means when these dry leaves, then cow dung and all these are exposed to bacteria, they get converted into a useful manure, that is also known as compost. Then in pharmaceutical, to make the antibiotics, bacteria are necessary. For antibiotics are streptomycin, tetracycline, okay, penicillin, these all are antibiotics. So these names of antibiotics are very important, keep in mind. This pharmaceutical industry, it is necessary for the metal recycling, means whenever any plant, animal or any organism is dead, all the nutrients whatever is present inside their body, they are taken out from the body and they are mixed in the soil with the help of bacteria only. If bacteria is not present, they will not be able to mix, or simply they will not be able to come out of the body, either of any body, living, any plant, animal, insect or whatever. So these are some of the useful effect of bacteria. Now coming on to the non-useful or harmful effect of bacteria. So in relation to the useful effect, there are some harmful effect of bacteria also. The first, you might have observed tetanus. All these type of diseases, tetanus, cholera, these all are different types of, due to the different types of bacteria. Clostridium tetani is the name of the bacteria or the causing tetanus. So in some of the animals also, in human beings also, it can cause some diseases. The most famous example is tetanus. Okay. Next is your, in case of plants, you will see that cabbage, there is rust of potato, rust of potato, rust of this sugar cane, and this in cabbage plant also there is some blight, okay, red blight disease, that is due to in the cabbage. In the plants also you can see so many diseases that can be able to, it is bacteria are able to cause many diseases in plants also. Next is your food spoilage, most important thing. When you will keep the food for some more time, due to the change in temperature, you might have observed the food gets spoiled and some smell starts to come out of that. Okay, it is called food spoilage, and when you will be eating that one, that will be called food poisoning. Okay, so disease is there, botulin, botulism is the disease. The botulin is the chemical which is formed in the food stuff when it is left over. Okay, food spoilage due to the action of bacteria. So this is the food spoilage, botulin, and botulism is the disease. Okay, these are some of the harmful and useful effect of bacteria which are very important, especially in pharmaceutical industries. The name of the antibiotics. Okay, so in the next class we will be starting with the protista, new important kingdom. Till then, thank you.